this video session, we'll be discussing about enzymes and its general properties. We'll be discussing every properties of enzyme. If we have to define enzymes, then we can define it as the biological catalyst that carry out thousands of chemical reactions which occurs in living cells. Or you can also say enzymes are the biological catalysts that speed up a reaction by providing an alternate pathway for reaction to happen. So the enzymes, they have an active site where the reacting molecules binds to, which helps in speeding up reaction. The enzymes are large protein molecules with three-dimensional structures. Each enzyme catalyzes a specific reaction or a specific type of reaction that involves a specific substrate. For example, they act as messenger between different cells, tissues, and organs. Now let us discuss about some general properties of enzymes. While talking about general properties of enzyme, the first point that comes in our mind is catalytic property, as enzymes are biological catalysts. So the enzymes are required in a small concentration for any chemical change. They do not initiate the catalysis but accelerate the rate of catalysis by lowering the activation energy. The enzymes remains unchanged at the end of reaction. That means you cannot visualize any kind of change in property of the enzymes at the end of reaction. And the other catalytic property of enzymes are they help to accelerate the forward and backward reaction to attain the equilibrium but do not shift the equilibrium. And the enzymes requires hydration for activity. Usually, the enzyme catalyzed reactions are reversible but not always. So in brief, you can explain the catalytic property of enzymes as enzymes catalyzes biochemical reactions. Small amount of enzymes have capability of converting large amount of substrate into product. So as a catalyst, enzymes increases the rate of reaction, but they do not affect the equilibrium of reaction. And the other important property of enzyme is colloidal nature. Enzyme molecules are colloidal in nature because of their large size. They are large in size, their molecular weight is high, hence the rate of diffusion is very low. They form colloidal system in water, and they are non-dissociable. That means we cannot separate them. And the third property of enzyme is protein nature. So the enzymes, they have all the property which globular protein have. All the enzymes are made up of protein, but all the proteins are not enzymes. The enzymes are high molecular weight compounds made up of chains of amino acid linked together by peptide bonds. And the enzymes can be found in many varieties, each functioning as a catalyst for different reactions. And the fourth property of enzyme is substrate efficiency. All the enzymes with absolute specificity catalyzes the reaction of one and only one substance. And enzymes with the relative specificity catalyzes the reaction of a structurally related substance. Let me give you some examples. Lipases hydrolyzes lipids, proteases splits up protein, and enzyme with stereochemical specificity catalyzes the reaction of only one of two possible enantiomers. For example, D-amino acid oxidase catalyzes the reaction of D-amino acid but not the reaction of L-amino acid. And the other property of enzyme is turnover number, or you can say enzyme efficiency. So the enzymes are extremely efficient catalysts, and some of the enzymes can increase the reaction rates by 10 to the power 20 times than that of the uncatalyzed reaction. So the enzyme efficiency can be defined as the number of substrate molecules change per unit time per enzyme. So the typical turnover number varies from 10 to the power 2 to 10 to the power 3 per second. For example, the turnover number for sucrase is 10 to the power 4. That means one sucrase molecules converts 10,000 sucrose into products. Similarly, it is 36 million for carbonic anhydrase, which is the fastest enzyme, and 5 million for catalase, which is the second fastest enzyme, and the enzyme efficiency is very low in lysozyme. And the other property of enzyme is regulation. The catalytic behavior of enzymes can be regulated. A relatively small number of all the possible reactions that could occur in a cell actually takes place because of the enzymes which are present. So the cell controls the rate of these reactions and the amount of any given products which are formed by regulating the action of enzymes. And the other very important property of enzyme is sensitivity. Enzymes are very sensitive to change in pH, temperature, and inhibitors. So first of all, let us talk about change in temperature. The optimum temperature of enzyme is 20 to 35 degrees Celsius. So the enzymes becomes inactivated at very low temperature and gets denaturated or destroyed at very high temperature, that is greater than 45 degrees Celsius. but the enzymes with low molecular weight are comparatively heat stable. For example, in Archaebacterium Pyrococcus furious, the optimum temperature of hydrogenase is greater than 95 degrees Celsius. This heat-stable enzyme enables Pyrococcus to grow at 100 degrees Celsius. And the second one is pH. The optimum pH of most endoenzyme is pH 7.0. However, digestive enzymes can function at different pH. For example, salivary amylase acts best at pH 6.8. Any fluctuation in pH decreases the enzyme activity. But sometimes change in pH causes the reverse reaction. For example, at pH 7.0, phosphorylase breaks down starch into glucose 1-phosphate. While at pH 5, the reverse reaction takes place. And the third one is inhibitors. Enzymes are also sensitive to inhibitors. Inhibitors are any molecules like cellular metabolites, drugs, or toxins which reduces or stop enzyme activity. Enzyme inhibitors are of two types, reversible and irreversible.